Greetings everyone and welcome back to Chapter 27. Last time we did a brief overview of the digestive system, and now we're going to delve in a little bit deeper and look at the histology and function of it, as well as what exactly it does. So the first question is, what is digestion? And digestion is acquiring nutrients from the environment. And we do this by what we call ingestion. So we take in foods and liquids, which will contain macromolecules, vitamins, and trace minerals and things that we're going to need. And we're going to use these large macromolecules, we're going to break them down into small molecules that can then be absorbed across the epithelial surfaces of the digestive system, and then they can be put into circulation where the body can use them. So we can either use these small molecules as building blocks for tissues and cells of the body, or we can use them for energy in order to fuel the ongoing processes within the body. Now, basically, digestion falls under the rubric of catabolism, and this is where we're going to decompose substances into smaller units in order to be able to use them either for energy or for building other substances. So we're going to need enzymes to do this because we're going to break down larger macromolecules into smaller molecules. So typically, by the time we have digested proteins, we will have taken them from their protein structure all the way down to small peptides and eventually into their individual amino acid monomers. If we look at starches and complex sugars, which are part of the carbohydrate group, we will break them down into simple sugars that can then be absorbed across the digestive epithelium. And when we look at fats, we can think of complex fats like cholesterol and triglycerides and fatty acids, and we can break these things down into smaller fatty acids that can be absorbed. So we'll look at that process a little bit later on. Obviously, we're going to require enzymes to do this, and we are also going to require oxygen, as oxygen is essential for cellular respiration. Now, once we have these small molecules, we can then use them as building blocks for tissues and cells and organs. And this would be the process of anabolism, which is a building up. Catabolism is a tearing down and is what basically digestion is doing. It's catabolizing or decomposing these larger macromolecules, larger molecules and substances into smaller molecules that can then be used to build up new structures and also for energy. And the anabolism is the building up part. So this is where we're using these raw materials such as amino acids, such as simple sugars and fatty acids to synthesize new compounds. The digestive tract basically is a hollow tube. It's called the GI tract or alimentary canal, and it basically runs from the mouth to the anus. And it will include the oral cavity, which is the mouth, the pharynx, which is the throat, which includes the the oral, and laryngopharynx, then the esophagus, which is a hollow muscular tube that runs to the stomach, then to the stomach itself, then to the small and large intestines, and the large intestine will end basically in the rectum, which ends at the anus. All right, so here we can have a look at it, and we can see here that we can start in the mouth, and we can see some of the structures in the mouth. And one of the things that we're going to know is that the epithelium that lines the lumen, or the interior hollow part of these organs, is going to be lined with an epithelium. And that epithelium is going to change depending on where we are. So in the mouth, we're going to have a stratified squamous epithelium. And that stratified squamous epithelium will continue down into the esophagus. And then it will transition into a simple columnar epithelium where we get into the organs that are going to be doing a lot of the heavy work of the digestion and later the absorption. So we're going to have a simple columnar epithelium from the stomach through the small intestine into the large intestine, and then all the way to the rectum, where it's going to transition back into a stratified squamous epithelium. Now, where we have stratified squamous epithelium is where we expect to see lots of abrasions, mechanical stresses. And in the mouth, we're going to see all kinds of chemical stimulants that could be abrasive or caustic to the epithelium. For example, when you eat lemon juice or coffee, you're going to be consuming acidic substances. or, for example, salad dressing with vinaigrette and what have you. So these things will also take their toll on the structures within the mouth, and so the epithelium has to be resistant to that. So we have a stratified squamous epithelium in the oral cavity, in the pharynx, and in the esophagus. And most of it is non-keratinized. However, where we have surfaces that are going to be involved in the manipulation of food, such as the dorsal surface of the tongue here and the hard palate here, we're actually going to have keratinized stratified squamous epithelium. All the rest of the epithelium in here is going to be non-keratinized, and we will see that the epithelium of the buccal part of the mouth, the oral cavity, which is the cheeks, is going to be continuous with the lips, and that's going to be non-keratinized stratified squamous epithelium. We'll see we have the tongue here, which is a very large muscle that has both a body and a root, and the root will be attached down here at the hyoid bone. And we're going to be able to use our tongue to manipulate things that come into the mouth and smush things around and mix the food with digestive enzymes that will be produced in the mouth, as well as fluids, water, this kind of thing. And that will soften this food bolus up into something that we can then swallow. So we have a couple of things in the mouth. We have teeth that will help us chew things up, and we have our tongue and our hard palate. And we also, that will help us with our mechanical breakdown of food, and that is to say, that's going to help us chunk large bits of food into smaller bits of food. And that will help it be mixed with enzymes a little bit more easily. It will create more surface area for enzymes to work on, and it'll also be able to mix with water and make a softer bolus, which is then easier to swallow. So if you've ever eaten Dorito chips, you don't want to swallow them whole because they would put a nice scratch all the way down your esophagus. Now, you'll notice we have some salivary glands here. We have 3 pairs of salivary glands. We have our parotid gland right here, which is just inferior to the zygomatic arch and really covers the posterior part of the masseter here. We also have our sublingual glands and our submandibular glands. And these are going to produce some digestive enzymes. And some of those digestive enzymes, for example, the ones under the tongue, are going to produce lingual lipase. And that's going to start the digestion of lipids in the mouth. We're also going to have something called salivary amylase, which is going to start the digestion of carbohydrates in the mouth. So we're actually going to start the chemical breakdown of food in the mouth, as well as the mechanical breakdown of food. Once we have chewed our food sufficiently and started or gotten that digestive process started, then we can go ahead and swallow the food, and it will travel first through the pharynx, which is the throat, and typically it travels through the oro and laryngopharynx. Hopefully it doesn't shoot back up in the nasopharynx. Anyone who's ever swallowed cola wrong knows what happens when the cola runs out your nose. Hopefully it won't do that. Hopefully it'll stay within the oro and laryngopharynx and then pass down into the esophagus, where it will then travel all the way down to the stomach. Now, the stomach is going to be one of the first places where we start digesting proteins. And the stomach has an enzyme called pepsin, which is going to digest proteins. And it also creates hydrochloric acid. And hydrochloric acid is important because what it does is it has a very low pH, obviously, creating a low pH environment, which will start to denature proteins. So as we ingest these large protein macromolecules, a lot of them will be enzymes from plants or meat products that we eat. We want to deactivate those enzymes so they're not active in our bodies. And we will also want to start to denature them so that our enzymes can act on them. It's much easier if we take apart the quaternary and tertiary structures of enzymes and pull them out into a long chain. Now we just have a chain of amino acids, and an enzyme can come along and chunk off each link of the chain bit by bit. So we can go one link at a time and start breaking down these amino acids. So that's where digestion of proteins begins, is in the stomach. We have very little absorption actually going on in the stomach. That's going to wait until we get into the small intestine here. And so the stomach will dump its contents into the small intestine, the first segment of which is called the duodenum. And the duodenum is going to receive the contents of the stomach. It will also receive bile that is made in the liver and stored in this gallbladder here, which contains concentrated bile. And it will receive secretions from the pancreas, which is an accessory organ that's making enzymes and buffers that will, for one, lower the pH, raise the pH of the contents coming from the stomach into the duodenum and bring them more toward neutral. And it will also secrete enzymes such as pancreatic lipase and pancreatic amylase, which will continue the work that we started in the mouth of digesting lipids and carbohydrates. Now, we will have most of our absorption happen in the second part of the stomach, the small intestine called the jejunum. And then we will have the ileum, which is the third part of the small intestine, and it will lead into the large intestine. Here we can see the large intestine. The ileum ends in what's called the cecum. Here we can see what's called the vermiform appendix here, which, as we learned in previous chapters, is actually a collection of mucosa-associated lymphatic tissue. So this is lymph nodes, a collection of lymph nodes right here, actually. And then, once it enters the cecum, it will travel up the ascending colon, across the transverse colon, into the descending colon, the sigmoid colon, and into the rectum. And it is here, at this area from the sigmoid colon transitioning into the rectum, we will see that the epithelium changes once again from a simple columnar epithelium into a stratified squamous epithelium. Because what's going to happen in the large intestine is we're going to take things that were not absorbable and we're going to dehydrate them and we're going to compact them for excretion. We're going to compact them into feces, which will then be stored in the rectum until they're ready to be excreted. And so at this point, we have a dehydrated mass of undigestible food or undigestible things that will then be excreted from the body. And once again, there's quite a bit of abrasion here. So this is why we have transitioned to our stratified squamous epithelium. All right, so let's look over here at some of our accessory organs. Once again, we have our salivary glands. That's going to start the breakdown of carbohydrates and lipids within the oral cavity. We have our liver, which is going to secrete bile, but it's also going to be a storehouse of nutrients, minerals, these kinds of things. And so all of the nutrients that we're going to absorb in the small intestine will go via the hepatic portal vein into the liver, where the liver is going to sort what we need right now. It's going to store any excess. It's going to release perhaps stores of things that we don't have enough of in our diet or we're not getting enough of right now. Say we get low on glucose, so the liver can release some glucose. But what we're going to do is we're going to store some materials here in the liver. We're going to sort stuff coming in from the small intestines and store it and release what is needed in the blood supply, with the exception of lipids. Lipids, as we know, as we have seen from previous chapters, are going to be absorbed via our lacteals, which are part of the lymphatic system. And we'll talk a little bit about that later, but basically the lipids will be packaged into chylomicrons, so protein-coated lipids that will then be absorbed into the lacteals, which are lymphatic capillaries. They will enter lymphatic circulation, and then they will join the blood circulation at the subclavian artery here, subclavian vein, where the lymphatic ducts will then empty into the subclavian veins. And then those lipids will enter general circulation, and then they'll find their way into the liver. All right, so we have our gallbladder, which is going to concentrate bile that is made in the liver. And the purpose of bile is to emulsify fats. So if you eat a big glob of fat, obviously we know that lipids and water don't mix. So we want to emulsify large droplets of fat into small droplets of fat, which is the purpose of bile. And that will then create, instead of one big ball, lots of little tiny droplets, which can be accessed much more easily by digestive enzymes. And so we will see that we're increasing the surface area by basically breaking down large globs of fat into smaller ones. All right, so we have the pancreas, which, as we know, is both an endocrine and an exocrine organ. In this chapter, we're going to be considering it as an exocrine organ and the enzymes and the buffers that it produces. So it will be producing pancreatic amylase, which will digest carbohydrates, pancreatic lipase, which will digest fats. It will produce several proteases, which are going to break proteins down as well, and it's going to put all of these digestive enzymes into the duodenum, which is that first segment of the small intestine, which will also be receiving contents from the stomach and the gallbladder. Now, the cells that are going to be excreting, or I should say producing, these digestive enzymes are located in the pancreatic acini. And these are sort of glands that end in blind sacs that have ducts that are going to end up going through the pancreatic duct, and as we'll see, they will empty into this duodenum. They will also secrete some hormones that we will see are important signaling molecules for signaling between organs of the digestive system to increase or decrease motility, or perhaps the secretion of enzymes. All right, so what are the 6 functions of the digestive system? Well, first of all, let's start with the mouth. We have to ingest nutrients. So we have to take in nutrients either by eating them or drinking them. And so ingestion is that very first step. And then we can start mechanical processing. And this is what we do with our teeth and our smashing things against the hard palate with our tongue, mixing up the food that we just ingested in with the saliva, which contains water and contains digestive enzymes and other things. And then we can start the actual chemical process, which is digestion itself. This is where the enzymes get to work and start breaking down large molecules into smaller ones. For digestion, obviously we're going to need secretion, because we're going to need to secrete these enzymes into the lumen of the digestive tract, wherever we are, and then these enzymes are going to interact with the large molecules and start breaking them down. Now, as we get into the small intestine, we're going to see absorption of these materials. We're going to see these really small molecules that we've broken down from larger molecules, and we're going to start bringing them across the digestive epithelium. We're going to put them into circulation. We're either going to ship them to the liver, or in the case of lipids, we'll put them into lymphatic circulation, and they will eventually end up being dumped into the subclavian veins, where they will enter into the general systemic circulation. Eventually, they will reach the liver, which may do further processing or storage. Finally, anything that is not useful to us will be excreted. So any waste products, any materials that we can't use. So for example, if you eat a plant and you have a lot of, for example, celery, and you eat stuff that cannot be digested, it'll be compacted and then excreted out through the rectum. All right, so we talked all about these things in detail already. So we talked about each one of these things is, and we will see that secretion is going to occur all along the digestive tract, starting in the mouth, and it's going to go all the way through, even into the large intestine, because we're going to be secreting things like water for mixing up with our food. We're going to be secreting acids, for example, hydrochloric acid, when we get into the stomach. Enzymes that are going to be breaking down the different macromolecules. Buffers, for example, bicarbonate, when we get to the pancreas. Pancreas will secrete bicarbonate into the duodenum, which will help neutralize some of that acid that came in from the stomach. Now, we will see that the stomach has a very thick mucus coat lining, and that protects the underlying epithelium and prevents the stomach both from digesting itself and also from giving itself chemical burns from the acid that it produces. Because basically the stomach, the hydrochloric acid in the stomach, is going to lower the pH down to about 1.5. So a very low pH, which is useful not only in starting to denature proteins, inactivate enzymes, but also kill possible pathogens that we might have ingested with the food that we ate. We're also going to see bile salts, which are going to be important for the emulsification of fats. All right, so absorption, we're going to start bringing in the important things that we need, the small molecules, the ions, electrolytes, vitamins, and so forth. And these will be brought across the digestive epithelium into the interstitial fluids of the digestive tract. From there, they'll either be brought into the capillary circulation and then into the hepatic portal vein to the liver, or in the case of lipids, as we've said, into the lacteals. Anything that does not get used will then be excreted, and basically the job of the large intestine is to remove the water, dehydrate them from all the stuff that we couldn't basically digest, couldn't use, and then package this up for processing and excretion. So this is the process of defecation, is when we have these bolus of dried materials that basically are not usable to us. Now, of course, we have to have excretion of or I should say secretion, all along these tracts of mucins and mucus to aid in the lubrication of these tracts. Because, as we say, there's going to be abrasion as boluses of food or boli of food or food materials or even indigested materials will be moved along this tract. All right, so we have to have safeguards against, for example, acids that we may not only ingest, but that we also produce. So as we said, the stomach has a thick coating of mucus, and it's going to be producing digestive acids like hydrochloric acid and things like pepsin, which are going to digest proteins. We're going to have mechanical stresses, such as abrasion. And we're going to be, as I say, going back to the example of the Dorito chip. If you've ever eaten a Dorito chip and chomped down on it really hard and had one of those sharp edges abrade your mouth, you probably know it hurts pretty much. But then within a very short period of time, you notice that cuts in the mouth heal very quickly. You'll also see burns. So, for example, if you have too hot coffee or your tea is too hot or whatever, usually you have a pretty good protection against overly hot foods. All right, also, we'll see that bacteria are ingested with food, and some of them are bad, some of them are not. So, for example, you've probably heard of probiotics, and these are bacteria that are good, and they actually have a symbiotic relationship with the host, and they actually help us digest our food. Without them, we would not be able to digest our food properly, and we'd end up with a lot of things that just don't get used. People who basically deplete the flora of their gut are unable to carry on efficient digestion. In the worst case scenario, it can actually cause death if it goes on to the point where somebody just has killed all the good gut bacteria in their intestines. So a lot of times what happens with an infection is bad bacteria will overrun the good bacteria and diminish them or basically crowd them out and prevent them continuing functioning. So a lot of times you probably, if you've ever been given antibiotics for a bacterial infection, a lot of times that will also have a negative effect on the gut bacteria as well. And so what happens after taking a course of antibiotics, people are often encouraged to take probiotics, which are the good bacteria that will then re-inhabit the intestinal walls and start doing their job of helping with digestion. All right, so let's talk a little bit about the histology of the digestive tract. We've already talked about the fact that we have an epithelium that covers the whole thing, that lines the lumen of the whole thing from the mouth to the anus. And that epithelium is usually either stratified squamous or it's simple columnar. In areas where we see high abrasion, we have stratified squamous, and in areas where we have more absorption and digestion, we're going to see simple columnar. But we have four major layers. The mucosa, which is comprised of the epithelium plus an underlying lamina propria. And included in that, we have two small muscular layers of smooth muscle, one that is concentric rings around the lumen, and the other that goes longitudinally along with it. And this is called the Muscularis mucosa. Underneath the mucosa, we have some dense, irregular, fibrous connective tissue called the submucosa. I should also point out that the lamina propria of the mucosa is an areolar connective tissue. Both of these, both the areolar connective tissue submucosa, the lamina propria of the mucosa, which is the areolar connective tissue, that's going to provide passageways for blood vessels and lymphatic vessels and nerve endings and so forth. It's also going to supply the nutrients of the epithelial layers overlying it. The submucosa is the dense irregular connective tissue that underlies the mucosa, the entire mucosa, and we'll take a look at a picture of it in just a minute. And it also provides a route for blood vessels and lymphatics and so forth. Outside the submucosa, we have something called the Muscularis externa. And the Muscularis externa is very important because, like the Muscularis mucosa, it's two layers of smooth muscle, one that's arranged in concentric rings around the lumen and the other longitudinally with it, but it's much, much larger. And this is going to be responsible for mass movements along the digestive tract, for churning and moving things back and forth within the digestive tract when we want to mix things up, and also for what we call peristalsis, where we take a bolus of food and move it along to the next segment. The Muscularis externa is important because it's basically going to be doing the major moving within the intestinal tract. The Muscularis mucosa is going to affect small areas of the digestive tract and local movements. Finally, outside the Muscularis externa, we have the serosa. And if you remember when we talked about serous membranes, you have the visceral layer, which basically you have an areolar connective tissue layer, and then you have a very thin, simple squamous epithelium that lines the inside of the serous cavity, and then you have the parietal side. The visceral side is flush up against the organ itself. Then you have the parietal side, which again, you have lining the serous cavity, that simple squamous epithelium, and then behind that, you're going to have your areolar connective tissue, and that's going to create this serosa or serous membrane. And as you recall, inside the serous membrane, between those two very thin layers of simple squamous epithelium, we're going to have serous fluid, which is a very slick, watery fluid that allows for lubrication and allows for the two parts of the serous membrane to slide a little bit, and it'll allow the organs to be able to move and churn and do their thing without creating unnecessary friction. So as we recall, most of the organs in the abdominal cavity are going to be covered in the peritoneum, which is a kind of serous membrane. And we will remember that the serous fluid that is produced within the serous membrane is a very thin, slick lubricating fluid that allows these organs to slide over one another. We will find that there are some organs that are not contained within the peritoneum, but are what we call retroperitoneal, that are between it and the back body cavity wall. And these organs we call retroperitoneal. And some examples of retroperitoneal organs would be the pancreas and that first part of the duodenum, as well as the rectum. All right, so let's take a look. Here we have a segment of what's probably the jejunum, or the second segment of the small intestine. And we will see that it has a mucosa, and the mucosa is lined with, or I should say covered, in this epithelium, this very thin layer on the top that's kind of pink here, that is simple columnar epithelium. Right below that, the yellow part, we're going to have the lamina propria. Then we have sort of the dark line here, that is the Muscularis mucosa. Then the sort of purple part here is going to be our submucosa. Then we have our Muscularis externa, and then we have our serosa. And here, as you can see, the inner layer is the visceral part, the visceral membrane. The outer layer is the parietal membrane. And so we have this double-layered membrane here. And as the double-layered membrane sort of wraps around the organ and comes out the other side, or I should say comes around the other side, the two parts of the serous membrane will fold together and create a mesentery. And so we will look at the mesenteries a little bit later on. You probably remembered them when we looked at our cat dissection and we opened up the small intestine. You could see this clear sheet that had these arteries and veins in them, and you could clearly see the arteries and veins. And basically, that's where we have the serous membrane basically coming together to form a double layer of serous membrane with areolar tissue in between and a nice passageway for blood vessels and so forth. Now let's look a little bit at this picture, because as I say, this is probably going to be in the jejunum somewhere in an area of the small intestine where we have a lot of absorption. You will notice these circular pleats. These are called plicae circularis, and they are there to increase surface area for absorption and secretion. You will also notice that we have these villi. And these villi are there to increase surface area for absorption and secretion of enzymes. Absorption of nutrients, secretion of enzymes. If we were to look very closely at the apical surface of the simple columnar epithelium, we would find that those cells have microvilli. So basically, we've made this massive increase of surface area by having all of these folds and indentions. And this is very important. This is actually what starts to flatten out in celiac disease. So people with celiac disease will lose a lot of these plicae circularis and their villi. All right, so as we've said, the mucosa is the lining of the digestive tract. It's what surrounds the lumen We have our epithelium. We're going to have all kinds of glandular secretions, not only enzymes, mucins, and other things that are going to help lubricate. And as we've said before, the mucosal epithelium is either going to be stratified squamous, typically, or it's going to be simple columnar. And the stratified squamous, we're going to see where we have areas of lots of abrasion, and the simple columnar is what we're going to see when we have areas of either secretion, absorption, or both. The epithelium, of course, covers the underlying lamina propria of areolar connective tissue, and that's going to provide a route for our blood vessels, lymphatics, and even some sensory nerve endings. All right, so as we've said, we're going to see the oral cavity, that is the mouth, the pharynx, which is the throat, and the esophagus. They're going to be experiencing all kinds of mechanical stresses, and these are going to have our stratified squamous epithelium. We're going to transition into our simple columnar epithelium with lots of mucus cells. You probably remember our goblet cells, which were chock full of little granules that have mucins in them, and when those mucins are expelled onto the surface, they combine with water to form mucus. And this is going to help lubricate the lumen so that boli, plural of bolus, of food moving through is not going to scrape and cause damage to these cells. All right, so along the digestive epithelium, we also have what are called enteroendocrine cells. And these cells are going to be producing hormones. And these hormones are going to coordinate the activities of the digestive tract along with accessory glands. All right, so as we've said, we've already looked at the plicae circularis. And we're going to see those in certain areas of the digestive tract, especially in the small intestine. The lamina propria, as we've said, this is our areolar connective tissue. It's going to provide a route for blood vessels, sensory nerve endings, lymphatic vessels, some smooth muscle that's going to be right there in that muscularis mucosa. And we're going to have some lymphoid tissue in there as well. All right, so we've already talked about the muscularis mucosa, those two concentric layers, one of which is rings around the lumen, and the other is going to create that longitudinal muscles that are running longitudinal or parallel to the tract. In areas such as the stomach, we'll actually see another extra layer that runs obliquely. All right, so let's have a close look. Now we're zooming in on one of those plicae circularis. So here's, you can imagine this going all the way around the entire lumen. And here we can see this part here, this is our epithelium composed of simple columnar epithelium with microvilli. Here is our lamina propria, this yellow stuff. We can see here some blood vessels. There's some glands here. We can also see, well, we don't see any nerve endings in here, but there are probably some nerve endings in here, some sensory nerve endings. And we know that this is areolar connective tissue. Then we have our muscularis mucosa with our rings going around the lumen versus the muscles that are arranged in a longitudinal fashion. Then we have our submucosa, the purple part. Here we have a larger gland that empties out onto the surface. Here we have some blood vessels, we have some arteries, veins, some lymphatic vessels, and we also have some nerves. We're going to see that we have nerve plexi, collections of nerves, and the digestive system has its own enteric nervous system. And the enteric nervous system is a standalone nervous system that can pretty much control all the actions of the digestive system. However, the enteric nervous system is closely tied into the autonomic nervous system of the central nervous system. And so we're going to have lots of parasympathetic fibers that are going to be intermingling throughout here. And these are going to stimulate functions of the digestive system. So remember that our parasympathetic nervous system is the rest and digest division of the autonomic nervous system. So it's going to enable us to stimulate the digestive tract, to stimulate enzyme production and breakdown of foods. Now, on the other hand, we're going to have some sympathetic fibers coming in here, and they're going to do exactly the opposite. They're going to shut down the digestive system, basically slow the actions of the digestive system, because when the sympathetic nervous system gets alerted, or I should say when we're aroused and our sympathetic nervous system starts firing, that's usually because we have a fight or flight, maybe life or death situation. And so all the important energy reserves and so forth are going to be dedicated to things that are going to help us fight or fly. So the digestion in that case will basically be shut down, and then we get to a time when we don't have to worry about some life-threatening situation, and then the rest and digest nervous system will take over, and then we can start our digestive functions once again. All right, so as we said, the submucosa, just a reminder, is dense irregular connective tissue, and it's going to surround that mucosa, and the outside of the mucosa obviously is the muscularis mucosa. We're going to have larger blood vessels and lymphatic vessels in here, and we're going to have some exocrine glands that may secrete buffers and enzymes into the digestive tract. As we said, we've already talked about the Muscularis externa. We've got our inner circular layer and our outer longitudinal layer. This is going to not only move stuff along the digestive tract, but it's going to be involved in mechanical processing as well. Sometimes we're going to have churning back and forth along segments of the digestive tract that acts like a mixing bowl or putting stuff in a mixer, and then it'll all be kind of mushed together back and forth in different segments of the digestive tract so that we can basically mix the contents of the bolus with more enzymes, and we can sort of help the digestive process along by mixing enzymes with the contents of the bolus. And then once we have done a lot of mechanical processing and we're ready to move on to the next segment of the digestive tract, then we have our peristaltic movements, which are going to push the bolus on to the next segment. And we have a lot of this is going to be coordinated by the enteric nervous system. So at the outer edge of this, as we said, we have the serosa, and the serous membrane is going to cover the muscularis externa in organs that are surrounded by the peritoneum. In the oral cavity, the pharynx, the esophagus, and the rectum is going to be retroperitoneal, the esophagus is going to be retroperitoneal. We're going to have, for example, all along the esophagus and the pharynx, we're going to have an adventitia, which is basically collagen fibers. So it's a sheath of collagen fibers, and this is going to attach these parts of the digestive tract to their adjacent structures. All right. So when we move things along the digestive tract, we're going to have these smooth muscle. We call them visceral smooth muscle because they're associated with the digestive tract. And they have what are called pace setter cells. And these pace setter cells will undergo spontaneous depolarizations that can be triggered by any number of things, such as stretching of the luminal wall, such as the presence of maybe a low pH or certain macromolecules. So basically, we're going to have contractions of visceral smooth muscle all along the digestive tract that will assist both in mechanical processing and in moving things along in peristalsis. So we're going to have some spontaneous waves of depolarization that's going to trigger waves of contraction through an entire muscular sheet. These obviously are set by our pace setter cells that we just talked about. We have them both in the muscularis mucosa and the muscularis externa. So peristalsis is when we move a bolus along through the digestive tract. And that happens basically well, here we have a good picture of it. Here we have our circularly arranged smooth muscle. You can see we've cut through them here, so they're coming at you from the page, and then we have our longitudinal smooth muscle on the outside. And so what's going to happen is we're going to have the constriction or contraction of the smooth muscle behind the bolus, and then we're going to have the constriction of the longitudinal smooth muscle ahead of the bolus, and that's going to cause the bolus to be constricted and elongated, and then it will be pushed along and moved forward by waves of these peristaltic contractions that move it forward. All right, as we said, some movements don't actually move the bolus forward, but move it back and forth within an area, a couple of segments. And this is called segmentation, and this is so that we can churn and mix fragments of the bolus with our intestinal secretions, such as our enzymes and fluid. And this is going to be sort of like turning your blender on and off or moving a mixing spoon about in your bowl randomly, so that basically we're just trying to mix the contents more thoroughly. All right, so let's talk a little bit about the control of digestive functions. We have both local factors, and these will include all kinds of signaling molecules. We're going to have some prostaglandins, histamines, and other signaling molecules that will be released into the interstitial fluid. This is kind of like paracrine signaling. They're going to signal cells that are close to them. They're going to affect adjacent cells within the small segment of the digestive tract. And this is going to help coordinate responses to changes in conditions like local pH, chemical changes, physical stimuli, stretching, and so forth. And this basically is going to affect a small portion of the digestive tract. The neural mechanisms, we've seen the enteric nervous system already. That's going to help move materials along the digestive tract, going to control some secretory functions. We're going to have the enteric nervous system, which works hand in hand, or I should say is stimulated by the parasympathetic nervous system, which is part of the autonomic nervous system, which is controlled by central mechanisms. All right, we have short reflexes in our neural mechanisms. These are going to be entirely enteric nervous system. They're going to be outside the control of the central nervous system. And this is going to control small segments of the digestive tract. Just like we saw earlier with our paracrine signaling, back here we're going to have local factors which are going to affect a small portion of the digestive tract. We're also going to have long reflexes which are going to involve central mechanisms. So this is what's going to cause our large-scale peristaltic waves. This is going to coordinate higher level control of digestive and glandular activities so that things are coordinated. And we'll see a little bit later that we have several phases of digestion, one the first of which is called the cephalic phase. When we think about digesting food, or think about eating food, our digestive tract already starts to prepare for the food coming in anticipation. And so it's going to have to start making enzymes and buffers and these kinds of things. We're going to have, once we have the food in the digestive tract, then we're going to have a lot of our longer reflexes controlling movements through the digestive tract in some of these peristaltic ways. All right, the hormonal mechanisms are also, these are sort of longer signaling molecules. We've got 18 peptide hormones that we know of, at least 18 peptide hormones that are going to be released by organs in the digestive system that will affect other organs within the digestive system. And these will be released into the bloodstream, and they will find their target organs, and they will signal them to do things like increase or decrease their motility, or increase their secretion of digestive enzymes or acids, or these kinds of things. These are going to be produced by our enteroendocrine cells within the digestive tract.